A very warm welcome to our esteemed guests. Good morning to our audiences in the US, and a very good evening to the audiences joining us from Europe and Asia. I'm Malvika, the moderator for today's conversation. Thank you so much for each and every one of you for joining us live today. This is the sixth episode of Biotechnology, an ongoing series of conversations with experts in the biotech industry. Here, guests share with us their thought leadership, their views on how the industry has evolved, their journeys and their perspectives on emerging trends and upcoming challenges. This is also part two of the series, The Journey of Oral Insulin, the Oromed Story. In the last episode, we spoke to Mr. Nadav Kidron, CEO of Oromed Pharmaceuticals, where he talked about the journey so far and the vision forward. In today's session, the panel will explore the science behind oral therapeutics in greater detail and its applications. On the panel today, we are proud to welcome Professor Avram Hershko, Nobel Laureate in Chemistry,
Hello.
2004, Dr. Miriam Kidron, CSO at Oromed Pharmaceuticals, Dr. Nupur Mehrotra, co-founder and COO at Premise Biotech, and Dr. Prabhuda Kundu, co-founder and managing director at Premise Biotech.
Yeah.
The format remains the same. After the panel conversations are over, the session will open for 20 minutes for taking questions from the audience. I request all the audience members to kindly keep dropping in your questions in the chat box below. As many questions as possible in the time that's available will be taken up and shared with the panelists for answers. Now I request Dr. Kundu and Dr. Mehrotra to share opening remarks and to welcome our guest speakers for the day.
Thank you very much, Malvika. A very, very warm welcome to Professor Avram Hershko and Dr. Miriam Kidron. And Professor Hershko, may I say how honored we are to have all of you, from all of us here, to have you on the panel today. I'll make a quick introduction, sir. So, for the audience, Professor Avram Hershko was born in Hungary and moved to Israel in 1950 with his family. He gained his MD in 1965 and PhD in 1969 from the Hebrew University, the Hadassah Medical School at Jerusalem. After a postdoctoral fellowship at the UCSF, he joined the faculty at the Haifa Technion, becoming a professor in 1980. He is now the distinguished professor in the unit of biochemistry in the B. Rappaport Faculty of Medicine at the Technion. Professor Hershko and his colleagues showed that the cellular proteins are degraded by a highly selective system. This system tags proteins for destruction by a linkage of protein called ubiquitin, which had previously been identified in many tissues, but whose function was previously unknown. Professor Hershko was awarded the Nobel Prize in Chemistry in 2004, jointly with his former PhD student, Aaron Shaikhnova, and his colleague, Erwin Rose. Amongst his many honors include the Israel Prize for Biochemistry in 1994, the Gardiner Award in 1999, the Alfred Sloan Prize in 2000, the Lasker Prize in 2000, and the Wolf Prize for Medicine in 2001, and the Louis Gross Horwitz Award in 2001. So with that, Professor Hershko, a very, very warm welcome to you, sir.
Thank you. Thank you, Prabhuda.
Alongside, we welcome Dr. Miriam Kidron. And Miriam, she serves as Oromed's Chief Scientific Officer and Director of the company since its establishment in 2006. As a pharmacologist and biochemist, Dr. Kidron earned her PhD in biochemistry from the Hebrew University of Jerusalem. For close to 20 years, she has been a senior researcher in the diabetes unit at Hadassah Hebrew University Medical Center in Jerusalem. She also earned the Bernd Schangler Award for her work on the diabetes research. She was formerly also a visiting professor at the medical school in University of Toronto. She's been the member of American and Israeli Diabetes Associations as well. A very warm welcome to you, Professor Hershko, Dr. Miriam Kidron. We are absolutely delighted and honored to host you both on this platform, Biotechnology.
Thank you.
Thank you, Nupur. So to open the session, Professor Hershko, I think our audience would like to get a brief, before we get into our topic, I'm sure our audience would like to know how you got to know about your Nobel Prize, sir, because there is a buzz that, did you receive a phone call? Did you receive an email, sir? How did it all happen, sir? We would love to know about that.
Well, the story is I never got a phone call. And the reason was that on that day, when they announced the prize, it was October 10th, 2004, there was a Jewish holiday, the Sukkot holiday. So everybody had a holiday. They tried from Stockholm to call the laboratory, but nobody was there. They tried then to call my home. They had these 2 phone numbers. They didn't have a cell phone of everybody at that time, but they had these phone numbers, so nobody was home. And the reason we weren't home, because we took 4 granddaughters to a swimming pool to have a good time. And they were small then, aged 4 to 10, all of them daughters. Yeah. So we had a great time there. And when we tried to get out of the swimming pool thinking to go home, I got a phone call on my cell phone from my cousin who heard it on the radio that I got the Nobel Prize in chemistry. And then my wife got on her cell phone. So we went home, and outside of our home, there were already journalists, and we were not looking very respectful. We were in swimming trunks, and but anyhow, we had a press conference then right there at our home because the Technion was closed. And only later I found on my phone a message, "Call back Sweden." So that is the story.
Absolutely.
So when they couldn't make the phone call, they released the news, and others got the news.
I'm sure it must have been an amazing time for you at that time. I mean, did you feel that elation?
We had a good time with our granddaughters. So I like to remember that.
Thank you so much.
Miriam, this would be a question to you then. How did you get to meet Professor Hershko, and how did this journey of working together start?
So actually, we got to meet Professor Hershko's student. He had a lot of students, of course, and he still have a lot of students. But one of his students that I know him, and he was kind of involved in our formulation, in our work. So he told me, listen, you need to get Professor Hershko to be one of your consultants. So we spoke with him, and I have to admit, I don't know if you remember it, Professor Hershko, but you said, I have to say that Professor Hershko does not do a lot of consulting. He does not like to do it so much. That I have to admit, and I am very proud that he was willing to come into our company and help us in the development. And later on, if you will want, I can elaborate on it. But when we met and we told him what we are doing and how we are doing, I always remember that he said, Miriam, this is the way to give oral protein. I don't know if you remember it, Professor Hershko.
Yeah, I do.
And this, I always quote it because for me, it was the one compliment in a lifetime. And that's how we met. And since then, Professor Hershko helped us a lot in development in the formulation.
Wonderful.
If I may add,
Sorry, Professor.
I had a secret diabetes connection. I never practiced, so I never had a diabetes patient. But I taught for many years medical students biochemistry on diabetes. So I knew quite a bit about the molecular basis of diabetes. So that was one of the reasons that I was interested. And of course, I understood the importance of oral administration of insulin instead of all the agonists that diabetic patients go through. Yeah.
Wonderful. It's absolutely heartening to hear this story. And do you remember, Miriam, which year was that? Or
no, I don't remember.
Okay.
But it was many years ago. It's over 10 years, no?
Yeah, about 12 years now.
I'm sure.
12 years already, yeah, many years ago. It's the very beginning. And actually, this is a connection to you, to Primas, because one of the materials that we are using, the SBTI, in which Avram are help us in the past and helping us now, was manufactured, the first time was manufactured by a different company. And I don't know, Prabuddha, if you want to tell the story, how we met. And Professor Hershko, he's the one that brought us to the real formulations that we are using now, using what you are manufacturing to us, the SBTI, the proper SBTI that we are using in all our pipeline, not only in the insulin.
Miriam, I can bring it up. I will bring it up a little later, but let it continue. I think we have lots of stories between ourselves, but I'll let Nupur continue now.
Okay. Thank you, for sure. I remember the stories very fondly as well. But absolutely, as Professor Hershko said, Miriam, this protein oral delivery technology, something which you call as PODS, it's a very powerful tool. Would you be able to tell us and all our audience, how did you get to the concept of it and how did you develop it?
So as you know, any science is not by one person. It's a team. It's a teamwork. And as you mentioned before, I used to work in Hadassah Hospital in the diabetes unit, and there we try to give oral insulin for many, many years with more success or less success. But one day we came to the idea that actually we need anti-protease to save the protein that we are giving. At that time, it was insulin. And we started with few anti-proteases, and we came at the end. We have done a lot of work, preclinical, clinical, to find the right anti-protease that will be good for human, because there is something that is a species specificity, that every species needs their own anti-proteases. And it's a lot of work to come to the correct formulation like we have today. And that's the way we have done, and with the help of Professor Hershko, of course.
Yes, I am going to extend this question to Professor Hershko. So how do you see the role of protease inhibitors or anti-proteases in the delivery of oral proteins and peptides?
It's clear that when you try to give a protein or a peptide orally, there are two major problems. One is that it will be destroyed. It will be destroyed in the stomach by the acid, by the acidity, by the protease, the major protease that is in the stomach, pepsin. And if it escapes the stomach, it will get to the small intestine where it will be destroyed by proteases that are secreted from the pancreas, like trypsin and chymotrypsin. And whatever is not destroyed will have a problem to get absorbed through the intestine. So that was the reason why insulin and other protein drugs were given by injection, which is a big problem when a drug like insulin has to be given every day or has to be given by pump, which also has its problems. So obviously, we try to overcome these difficulties. Now, the difficulty in the stomach that was done before me by Oramed, it was done by a capsule that has a coating which will allow it to escape the stomach, enteric-coated. So the insulin is within a coated capsule. The capsule will not open in the stomach. It will open only the more basic pH of the small intestine. So that takes problem of the stomach. Now we got to the intestine, and we have these problems of destruction of a protein and adsorption. Adsorption, there are enhancers of absorption, such as EDTA in this capsule. There are others, and it works quite well. So that will take care of the absorption. But the destruction was a big problem. And when I got to meet Miriam for the first time, they already had the idea of using proteinase inhibitors from from soybean, which were available at that time. You could buy them from Sigma Chemical Company as a mixture of two inhibitors. One is an inhibitor of trypsin called Kunitz trypsin inhibitor, or KTI, and the other is an inhibitor of both trypsin and chymotrypsin. It is called Bowman-Burke inhibitor, or BBI. It is a two-headed inhibitor. It has a site to inhibit both trypsin and chymotrypsin. So when I got there, they tried to use that, but they had each batch of what is called SBTI, soybean trypsin inhibitor, which is a mixture of these two. Each batch had different problems. Some worked and some didn't work. So with my former student, as you mentioned, Arya Meir, Miriam noted that there was a correlation that those batches that didn't work well had less BBI. They had KTI and the KTI was active, but they had less BBI. And actually BBI is very important because it inhibits the other major protease, chymotrypsin. So if you inhibit one, it's not enough. You have to inhibit both to let the insulin escape. So I got to that, and because BBI is present in small and variable amounts in soybean extracts, it was clear to me that we have to work out a method for the isolation, purification of BBI, and then to add it to KTI at the desired amount. And that's what we did. It took quite a while, but we worked out the procedure, and that's the procedure that's now being used. There are other ways to go about it. If you want to talk about it later, Miriam. But I think that was the major development, major improvement that allowed this capsule to be effective. I want to add something.
No.
Yeah, no, I think what with the help of Professor Hershko, we were able to come, I think, to the optimist formulation. There is always maybe possibility to do, but we have to remember what I mentioned before, because of the specificity of the antiprotease between animals, any kind of animal, and human being like an animal. And so we need, if our target is human, we need to do always the final target is a clinical study, which of course it's not so simple, it's expensive, and everything which comes with any clinical study. So I think that what we have is a very good formulation.
Thank you, Miriam, and thank you, Professor Hershko. Miriam, as Professor Hershko indicated, it provides a very safe, efficient way of delivering insulin. Now, moving a little towards the one that you first tried with, you think that it's by mimicking the body's natural process of the insulin going directly to the liver rather than the bloodstream? Can you elaborate it on for our audience as to how the impact is going?
Absolutely.
A little bit about
sorry. Avram, okay, go ahead. Go ahead.
I go to medical students, so I know about it. Insulin works on different tissues like the muscle, the adipose tissue. It also works on liver and inhibits the production of glucose in the liver, and which is very important, especially at night when the liver produces a lot of glucose. So normally, here you have to know a little bit of anatomy. Insulin is secreted from the beta cells of the pancreas, goes into the blood, and whatever is going out from the pancreas goes first to what is called the portal circulation that goes through the liver. So the insulin sees the liver before it sees all the other organs, and only whatever escapes the liver goes through the circulation of the liver, escapes the liver, acts on other tissues. Now that's very important because the action of the insulin, when you inject insulin, doesn't go to the liver. It goes to all the circulation, and only part of it gets to the liver. So here, because whatever is ingested, and this whatever is not ingested, whatever is going to our gut, and then is absorbed from the gut, also goes first to the liver by the portal circulation, and only then to the rest. So here, giving the insulin capsule, the Oramed capsule, actually mimics the way the physiology in which insulin meets our body. First goes to the liver and then to the rest. Now, this is very important, especially at night. And we actually give the capsule in the evening, because then it will work well at night. Because at night, we have a lot of glucose production by the liver. And the insulin, oral insulin that goes directly to the liver is present there at high concentration and will inhibit the production of glucose, and therefore the elevation of glucose levels during the night. And that's a very important part of the treatment.
And may I add, with your permission, Avram, may I add only one point, is that the pancreas produce more insulin. I mean, the liver sees more insulin than the circulation. The liver can take care of over insulin. It sees like, I think it's more than 2 times the insulin that needs to get to the circulation. The liver use the insulin for what it's doing, the gluconeogenesis, everything. They're using the insulin, and there is a special enzyme that cut off the excess of insulin. And due to it, what Professor Hershko said, that when we use Oramed insulin, oral insulin, we are not afraid of hypoglycemia, because the first pass is the liver, and we know that the liver can get around with a high amount of insulin. And that's why we have shown in our clinical studies that we do not have a hypoglycemia effect, which is very, very important when giving insulin.
Well, thank you, Miriam. Miriam, when we extend this technology to other therapeutic areas using the POT technology that you referred to, what are the other therapeutic areas or modalities that you think, apart from oral insulin, that you are trying or will try immediately?
Yeah, I always say that Oramed is a platform technology. It's not only for insulin. I mean, it's a platform technology for proteins. We have not done studies with proteins that are higher molecular weight than insulin. All the studies that were done, they are less than insulin. But I am pretty sure that with the help of Professor Hershko, we will be able to overcome higher molecular weight of protein. So we are working with GLP-1 analog. We have done few studies, 2 studies in Israel and 1 study in the US. We have an IND in the US for the GLP-1 analog. We have done in healthy volunteer in the US. In Israel, we have done also on type 2. And now we are in a process to start same study on type 2. The results in the first studies that we have done, the results are very encouraging with this GLP-1 analog. And we do hope that we will do it with type 2, that we will have a very good results. Then we have the leptin. The leptin, which I have to mention that we were able to get from Primas. The Primas manufactured it for us, and we are thankful for it. And I do not want to go and elaborate about leptin, but we have shown in preclinical that when we dose with leptin using the same SBTI as we have with other protein, we were able to show in pigs PK results. Now we are towards the end. We have done a study. We don't have yet all the results with a type 1 because, of course, to use leptin for obesity, we cannot do it in a short time. So we decided to do it on type 1 to show there is in the literature, if you give leptin to type 1, you can show reduction of glucagon. So we have done a first study here in Israel on type 1 with leptin. We are waiting for final results. It was a preliminary study. I don't think we will have PD results, but it was important for us to show the safety issue. So far with the safety issue, I know that we do not have any safety issue with it. And we will see once we will have the results, how we will proceed, how many people, etc. But it looks promising, the leptin results. I hope it will be a good molecule for it. But of course, it's a long, long way. Long way.
Thank you, Mary. Go for it.
Absolutely, long way, Miriam.
Some other uses, maybe anything that goes with for small molecular peptides, such as growth hormone. It could be a good way to introduce it to children who need it. I think it may have a good implication in vaccination, because actually for vaccines, you don't need a big protein. You need small peptides that are presented to the immune cells. And if we can prevent their degradation in the gut, they will go, and it will be good, for example, in third world countries where injection may be a problem, or now you hear about the COVID thing to transport these antigens, where it will be very difficult. But to give a capsule containing already peptides which are antigenic might be a good solution. So there are all kinds of solutions that you can see, all kinds of uses that you can see other than in diabetes.
Yeah, thank you, Aaron, for mentioning the vaccine. I always said that it will be much better to ship everyone into home a capsule in an envelope, rather than come to the clinic and have an injection. You swallow it and there will be much, much less side effect, no side effect with it, and it will solve the problem of the world. But we have to do a lot of work with it. Yeah.
Absolutely. I think we're all encouraged, and we do want to see the results, as you said, Miriam and Professor Hershko, that there could be an application in multiple areas. But if I recount back a little, few years back, Miriam, and if I could ask, you must have faced a lot of challenges when you were looking at the results and developing it. Is there a few examples that you could share with us and our audience on how some of the challenges looked at, how they looked at you and how you looked at them, and how you and Professor Hershko and the team, you all got together to solve them?
So I can start, and then Professor Hershko can go ahead. I can start from the very beginning. When we moved from preclinical to clinical, as I said, we did not know how much insulin to give to human. We did not know how much antiproteases to give. This was before the time that we knew Professor Hershko. And we have done, and each study was a huge burden on the company, and we did, and when we get the results, the first time that we have done it in clinical study under Oramed in Hadassah, we did have some good results, but we switched a little bit. And the first, I have to admit, was not so good, because we learned a lot from it. We learned what should be done, how much insulin should we give, what will be with the SBTI. We started with 2 antiproteases. Today, we have SBTI, as Professor Hershko mentioned, it has 2 antiproteases in, like we can say, built in. It's not built in, but we call it SBTI, but it has 2 antiproteases. But then we had, beside SBTI, another antiprotease. And slowly, by only by clinical trial, we could achieve it. To the final formulation that like a year ago, we finished our phase 2 study, very good results. And now next week, we are starting our phase 3, which is, I think it's amazing for a company like Oramed. Starting a phase 3, and with the help of all of you, Primas and Professor Hershko, that help us to get to the good results in the clinical studies.
Yes, it's a big leap. I agree. It's a very big leap to get into phase 3. And Professor Hershko, as Miriam said, you've been involved in very many scientific challenges, and you've been a pillar of strength, pillar of knowledge for everyone. How would you look at some of the challenges which have come along the way and your outlook, sir?
Well, most of my life I'm doing basic science, and to develop a drug is quite different, yeah. And there were all kinds of challenges that I heard from Miriam, I didn't make the right capsule, the formulation, you have to have an oil and the correct oil. What is the formulation of oil? Things like that. And I even remember a preliminary clinical trial that didn't work because the capsule wasn't built well. So you have to take care of all kinds of things. Well, in basic science too, you have to take care of the details, but the details are different, yeah, different. And the organization of a clinical trial is, of course, different. So I was very pleased when the phase 2B worked so well. There was a good reduction of hemoglobin A1C and a significant reduction of blood glucose. So, it seems that we got over all the technical problems and also the theoretical problems of how to protect insulin and how to let it get absorbed in the intestine.
Thank you very much, sir. Miriam, now let me take us to a little bit different track. I know there are over 350 to nearly 360 people on the audience, and I'm sure they would all have one question, which similarly we had asked Nadav, is what made you think of starting the company? Where did that incentive come from? And then I'll come to a story which I would like to share.
But please, I could not hear your last sentence before you said the story. I could not hear the sentence.
I said, what made you start thinking of starting the company Oramed? How did you start that?
I don't know if the audience know, but Nadav is my son. And Nadav has always said that he grew up with oral insulin. As I told you before, I was for many years in Hadassah in the diabetes unit, and we tried oral insulin. But in a place like hospital or in a place even in university, you don't have the sources to go beyond what you achieve. And one day I told Nadav, listen, I think I'm pretty sure we have something in our hand, because it was at times that it was preclinical studies that we have done, and the results, the reproducibility was very good. One of the problems in developing anything, and maybe even in a basic science, is the reproducibility. Because if you get one time result something and the other time a different result, so what is the result? You don't know what is the true result. But here we got to a point where we had reproducibility with good results, a reduction of blood glucose in the dogs. At that time, we used dogs as preclinical. And Nadav, in his personal life, got in a situation that he took it and he decided to go and to see. And it was a lot of work, Hadassah had a patent, he needed to do it. And then, of course, Oramed, since Oramed came into reality, so Oramed have a lot of patents today in most of the world. And we achieved a lot. I think during the time since the company started as a small company, we are not that many people. We achieved a huge amount of everything that we have done, and I think it's nice.
No, thank you, Miriam. And I think this is the time I would like to tell this story for the audience. So Nupur and I were traveling to the US in 2013 around April to the BIO convention. And this was a first for all of us. We had never been to such a large conference. And when we walked in there were about these thousands and thousands of people and booths, hundreds of booths. And we set up ours in the India convention area, which the government of India and DBT had very kindly taken on our behalf. And we took a small place there. And Miriam came along, and it was around 11:55 in the a.m. And she came and she said she had to meet some other people there. And she had not come to meet us. So we were just incidentally there. And she said, where are they? And we said, we don't know. And so she said, okay, I'll come back. So she walked away and she came back again in 5 minutes for the 12 o'clock meeting, and the folks had not been there. So then we could see that she was obviously a little jet lagged and tired. So Nupur offered, why don't you sit down? So she sat down and said, and as you can all realize, she looked around and said, what do you do? And we said, we make proteins. She said, I have 2 proteins. And that's how this amazing journey started in 2013, where we are now partners with Oramed and making the material for phase 3. It just started by offering a seat, a glass of water, and I think it's destiny, Miriam.
Yeah, I think it's amazing. I think it's amazing our coincidence of meeting, and we were able to come from there. And I hope you don't mind if I will tell the story that when the first time I did not know if I have done a good stuff. I did not want to travel to India. And I did not know what, and everything was the beginning. It was via email, telephone, and so at the end we did travel, yeah, it was when we started from zero. I mean, Oramed and Primas, we started from zero, and we achieved a lot. We still have a lot to achieve. We still have a lot to achieve. And maybe it's a time here now, if you want, we can mention the RBBI, the recombinant RBBI.
Please.
Maybe Avram will tell about the recombinant.
Yes.
Well, as I said, the weak point in our formulation is BBI, because it is present in lower amounts in soybeans and the other compounds. We even looked at sunflowers, all kinds of inhibitors. So chymotrypsin inhibitors are a problem. And then the idea came that maybe it is better, and there are also differences in the activity in different batches. We buy the soy flour, and soy flour is made by the industry, and you have to be careful that it's not cooked. Usually it is cooked so that the inhibitors will be killed. You don't want that. But even without cooking, there is a big difference in different batches and big differences, different productions, different years of soybean. So it would be better to have recombinant inhibitors. Now, this is not a trivial thing, because these are extracellular proteins. They are secreted outside the cells. They have a lot of disulfide linkages. BBI has 7, these disulfides, so they have to be correctly formed. And it's quite a challenge to make them, but Primas did it, and now they have a very good active BBI, and we are testing whether it can replace effectively the natural BBI. It's actually more active, because evidently there are some post-translational modifications that decrease the activity. So the recombinant BBI is about 2.5-fold more active than the natural BBI. So it looks to be promising, and I think there is a good chance that it will work very well.
Thank you so much.
Mm-hmm.
No, so I just want to add on what Professor Hershko said. As I said, the final target is human, and we need to do now. I hope we will be able to do very soon, with the help of Primas, a clinical study to show that what we see in preclinical, our model is pigs, so to see if we get the same results that we are having in the pigs, and this will be a great moving forward for us, for both Primas and Oramed.
No pressures, Miriam, no pressures, that's fine. Yeah, Nupur, please continue.
No, absolutely. Thanks, Froda. Thanks, Miriam and Professor Hershko. I know RBBI is the next focus. But Miriam, again, as you mentioned, and Nadav also in his previous talk with Prabhuda mentioned that you've kept a small team, you've worked more with collaborations. Primas very happily and in a very honored way is one of the collaborators. But how have you walked through these multiple collaborations, and how have you taken it forward, and some of the highs and the lows? If you want to comment a little bit on that, I think it would be very helpful for some of the companies and other people who are setting up ventures to learn that from you.
I think collaboration with other people, companies, it's very, very important. This was your question? No,
It is.
I think it's very important. Nobody can do alone. Nobody knows everything alone. And you have to know where you need to look for advice, or you need to do, like, if I will give an example, like the leptin that we came from. We looked around and then we came to you, and this was the best offer that we can have, having a leptin, a human leptin, what we are having, correct? Yeah. So it's very important to everyone to know that you need collaboration, but it's important to find the right collaboration. You know, that people and companies that you get along, you don't have problems and so on.
Yeah, no, thank you. Absolutely well said. Further, yeah.
So looking ahead, I think you've also answered a few of those, but looking ahead, where do you think this will go, the oral technology? I know there are multiple companies who have, started working on oral therapy, and they have been for years working on oral therapeutics. Some of them have succeeded, most of them have not. And as Miriam correctly pointed out, that going into phase 3 is very important, but not the end of the road. And we have multiple other complications coming up. So in your view, if you were to address our audience and say, where do you think the future of oral delivery is going?
You mean for proteins, because we are dealing with proteins.
Yes, yes, of course. That's what I asked, Professor.
You mean for proteins? I think I think the world is ahead of. I mean, it's very important, and I am pretty sure. I hope we will have a success, of course. But we can see what Novo Nordisk have done. They have now they have a protein, an oral protein. And their proteins, they have the GLP-1 analogs. Their GLP-1 analogs, they have it as an oral protein. But I have it to say that, I don't know if it's good to say it or not, but our bioavailability is much higher than what they have. I think, maybe I should not say it, I don't know. But I think that our formulation is the much better approach for oral protein. And I think the world starting to understand that it is possible to have oral protein. Years ago, people say it's impossible to give oral. It's impossible to get oral protein. It's impossible. But you see that it is possible, and it's coming to reality. Like we are starting phase 3, and we have good results with insulin, and we have a lot of encouraging results with the other proteins that we have done clinical studies. So I think that the world will have oral protein. I don't know how long it will take, because we have to remember that the regulatory is a huge obstacle. Maybe not obstacle, but to fulfill all regulatory requirements, it takes a lot of time and a lot of resource. It's not like they're developing the COVID-19 vaccine that they cut off the regulatory. For a regular thing, you have to go, it's a lot of toxicology, a lot of preclinical, a lot of clinical studies until you get a proof of it. But Oramed, with the insulin at least, we are on the right way for it.
Coming to Professor Hershko, before we I think we are now coming towards more or less the end of our discussions. But sir, in your opinion, do you think there will be certain challenges in the pediatric side to take these formulations? What do you think the differences will happen in that, sir?
In juvenile diabetes?
Yes, sir.
Well, it is intended for juvenile diabetes. It actually works in juvenile diabetes. So I think it can be taken to at early times in children, of course, and it will save a lot of suffering for these children.
Thank you.
So I think we're coming to a place where we have a lot of questions from the audience, and I would like Malvika to bring that up. But before that, Miriam, if there are any concluding remarks that you would want to give, And then we can open the questions from the audience to that.
I don't know what kind of remarks you're expecting. As I said the oral proteins will be available, and it is available, and collaboration like we have Oramed and Primas is very important. All kind of collaboration can bring everything together and solve problems.
Yeah.
Thank you very much. And before I ask Professor Hershko to sum up and giving his concluding before we get into the question answer, sir, I don't know if you remember, we were sitting and discussing a small or rather large problem in the way we were estimating things at the offices in Jerusalem. And after a little bit of hard discussion where I was getting up on the board and doing the mathematics, I think you really blessed my, if I have a biochemical avatar or a biochemical life, you put your arm around me and you said, he understands biochemistry. I think coming from you, I think I've been blessed with that. And that was an amazing opportunity with you where you just put your arm on me and said, he understands biochemistry. I don't think anything else would have satisfied my biochemical life after that. I thought it's amazing. Sir, before I end, what would your message be? We have lots of people who are listening to you, to all the young aspirational scientists across the audience.
As I said, I've been doing basic science most of my life, and I thought I would continue like that. I wasn't involved in many companies, but I like this small company, Oramed, because of two reasons. Maybe they had the right attitude, the right people, and also, as I said, I had some connection to diabetes from teaching. I was teaching the biochemistry of diabetes to medical students. And I was actually, I worked on protein degradation, ubiquitin-mediated protein degradation. So the important part is the marking by ubiquitin. But then there is a protease. So I knew about proteases, and then I knew about protease inhibitors. So I got involved in a company because I thought maybe I could use my knowledge from basic science about proteases, proteinase inhibitors, enzyme purification, protein isolation, and I used all that from my knowledge in basic science for application. So basic science is sometimes you may get involved in such things. In development of drugs, as long as you don't do it too much, that's okay. I don't think one should do it full time. One should stay with the basic science. But to help out, I think it was an experience for me.
Thank you, sir. It's always an ever humbling experience to have you and Miriam on a webinar like this and discuss with you. You are both amazing people, and it's absolutely fortunate for us to even have to know you and know you personally. Thank you so much. Now we will open the platform for question answers. Malvika?
Absolutely. Thank you so much to everybody for this wonderful interaction. We definitely have a lot of questions that have lined up. I would like to start with the first question from Karthik Ramani, who asks, will oral insulin delivery eliminate injections or complement it? We need both mealtime and basal insulin to achieve glycemic control. What is the Oramed experience in achieving this?
Maybe Miriam, you want to answer that?
Okay. So actually the oral insulin is not to avoid the injection. Let's start with type 2. The idea in type 2 is to start the oral insulin before the injection of insulin. And by doing it, we will avoid at the end of the day of the injection of insulin, because we have to remember that even though today there is a variety of medication for type 2, but still at the end of the day, 50% of the people of type 2, they will need the injection of insulin. And taking oral insulin, it will avoid, excuse me, excuse me. And type 1, it will not be to avoid the injection, but it will help them in a better control of the diabetes.
All right, wonderful. Ghosh asks, is glucose production by the liver temporarily regulated in this case?
Is it regulated normally? Yeah. Well, that I can tell you from my lectures to medical students. Yeah, it is regulated by hormones, by insulin. It is inhibited by insulin. It is regulated by glucagon. It is stimulated by glucagon when there is a need normally for glucose production. So it is regulated by hormones, and the mode of the hormonal regulation is known. Yeah. The details, which enzymes are involved.
Right. Swaroop Bhattacharya asks, will this insulin be able to correct hypoglycemia?
Yeah, the answer, what does it mean to correct? We have seen in our clinical studies that we do not have hypoglycemia using the Oramed insulin, oral insulin.
To prevent hypoglycemia.
Yeah, yeah.
Right.
Absolutely.
In a gentler and more physiological way.
Right.
Yeah.
Kartik Ramani asks, what is the level of HbA1c control which has been achieved with the oral delivery of insulin compared to other ODA and injectable insulins?
I did not hear well I did not hear the question about hemoglobin A1c. What is the level of hemoglobin A1c that we are going to treat?
Yeah. What is the level of control compared to other oral deliveries and injectable insulins?
In our study, we have done study, we started, the people were from 7.5% hemoglobin A1c up to 12% hemoglobin A1c. In the phase 3, it will be from 7.5 to 11% hemoglobin A1c. We think that it will be good for all. What we have shown it in the phase 2b, that even people that have three medication of anti-diabetic, they are still on a high hemoglobin A1c, but adding Oramed insulin, oral insulin, we were able to reduce with more than 1% reduction of hemoglobin A1c. But today the target is to go below 7% hemoglobin A1c. And we will see in our phase 3, hopefully we will get to that target.
All right. The next question is from True Rogers. He asks, is it important to target certain sections of the intestine for maximal absorption into the hepatic portal vein for hepatic delivery of the insulin?
Avram, you want to answer?
We don't know much about it, and also it's not clear how can we achieve that, to target different segments of the small intestine. The current methodology is just to open the capsule, the enteric-coated capsule in the small intestine as the pH rises. To target it to different places, you would need some other controls. There are such things, but much more expensive. So we didn't think about it. And I'm not sure it's worth to go to all the expense. I'm not sure there will be a big difference between different segments of small intestine as for absorption. So the answer is, I don't know the answer.
All right. The next question is from an anonymous watcher who asks, any side effects of giving insulin orally along with protease inhibitors?
No, we have not seen. It's amazing. It's amazing the safety of this formulation. We have not seen, I mean, there is always side effect when you do a clinical study, of course, but there was no difference between the placebo and the active in the phase 2b, no different at all. And the serious adverse events that we had, all of them were non-related to the formulation, all of them non-related to the formulation. So it is a very, very, very safe formulation. We have to remember that the antiprotease that we are using at the end of the day, it starts from soy flour, it starts from a food. So we don't expect that there will be any problem with it. And we have not seen, not in the preclinical, not in toxicology, and not in the clinical studies that we have done so far.
I would like to add here that anti-proteases, it's very important to know how much you are giving. If you give a huge amount, then you will inhibit digestion of proteins, and then the patients may go into protein malnutrition. But that is 20-fold higher than the amount present in the capsule. The amount present in the capsule is released from the capsule together with the insulin, and just locally, it's that segment of the intestine inhibits the proteases, and for a short time, for a couple of hours. And then, so at that level, there was no, all the toxicological studies showed that there is no protein malnutrition whatsoever. So it's very important to know how much are you giving.
Malvika, is it possible for me to just jump in once? Because I see a lot, there are more than, I think, 50, 60 questions. It's going to be very difficult for you to handle. I can sum up one or two basic areas, if that's okay with you, and then, you could take it forward. Miriam, there's a lot of questions on the bioavailability. So questions, I mean, there are at least 7, 8 questions on bioavailability. There's a few questions on the amount you use. And is it a once a day or is it twice a day capsule? So I think this will take care of, sorry, Malvika, but it will take care of a lot of the concerns that are there.
So may I ask, if someone wants a specific question, I will be happy to answer by mail. But for your question, bioavailability relative to protein, giving oral protein, our bioavailability, which we have done in a small study in dogs. It's very hard to do bioavailability because we mention all the time that the majority of the insulin gets into the liver. So it's very hard to measure how much you can do liver perfusion. But with human, it's hard to do liver perfusion. So what we have seen, and we had a poster, Oramed had a poster a few years ago in the ADA, what we have shown that we have more than 5% bioavailability when we dose with our oral insulin. We are using now 8 milligram of insulin in our formulation. This will be our phase 3. It will be either once a day or twice a day. We have very good results with once a day, very good results with once a day in the evening. But we decided to go ahead with twice a day, maybe in a longer period of time, because phase 3 is for 6 months and 12 months. There will be a further, we will see further reduction of hemoglobin A1C. But so far, once a day, 8 milligram, it's a very good results. And what was the last question? You say there was 3.
No, in terms of, you've addressed it. One was bioavailability, the amount of insulin, and then it's a once or twice a day.
Okay, thank you. Thank you.
Please.
To take the therapeutics out to the market, what are the challenges and the hurdles that you faced in this journey?
We started with the US, with the FDA, and I have to admit they were very collaborative, and they I don't know, they treat us nicely. And of course, we advised, like we started now, we are going to start phase 3. We had the end of phase 2 meeting with them, and they gave us their advice about the protocol, which of course we accepted. And I mean, you need to do stuff. I mean, you need to do before you go to the regulatory, because you know what you need to do. You need to do toxicology. You need to do a lot of preclinical studies. You need to do a lot of clinical studies. You open your IND, and then you have to do. But the regulatory, the FDA, we did not have this specific challenge that we could not overcome.
Right. Okay. Suparna asked to the panelists, she says, how many participants, and I think Miriam, this is again for you, how many participants do you plan to enroll in the phase 3?
Around 1,200.
Okay.
So, Malvika, if I may just jump in, I think a couple of questions are coming on. When do you think, Miriam, you would reach to a level where phase 3 study data could be out? And I think a lot of expectation is when could be it available in market if things go well?
So in the FDA, it says that you have to finish the phase 3 and then you have to apply for your BLA, which in the case of insulin, So it will give us a 12 years advantage. Does not matter of our patent. It will be like unique for Oramed, this using of our formulation with the insulin. We hope that in 2 years, maybe it's optimistic view, that within 2 years or 2.5 years, we will have as a result of, we have 2 protocols for phase 3. And then it depends how long it will take to get the BLA. It's between half a year and 1 year. So it's between 3 years and 3.5 years, we hope to be there.
Thank you. Malvika, please.
Malvika, there is one more question which has just come flashed into my thing. It's from Dr. Rustam Modi. He addresses it to Dr. Miriam. Insulins have a longer circulatory half-life. Oramed's exenatide is in phase 2B, which is very short plasma. Does it use any different technology from the oral insulin? This could be a potential blockbuster like the Ribelsus. Good luck. So do you have a comment for Rustam on this?
I'm not sure I understand the question. I heard some because I have a problem with the internet use, so I'm not sure I understand. Can you repeat only the first part of the question?
I guess the question is that between what you're doing with exenatide and oral insulin, is it using the same delivery system?
Yeah, correct.
Okay, so that's
yeah.
All right, great. Malvika, over to you.
So we're actually quite close to the end of time. So I'm just going to take like 1 or 2 more questions. This one is from Ritu, who asks, will long-term type 2 diabetic be able to switch over to oral insulin?
I'm not sure I understand the question. I'm sorry. Ivan, did you understand the question? I didn't.
Well, as I understood it, in the long time, or eventually we'll be able to use only oral insulin. I think we talked about that. Hopefully, why not? But for that we have to improve the delivery, maybe to improve the absorption also, and so further improvements. Right now it is an auxiliary system. So we can control much better blood glucose, do with less injected insulin, do with less other drugs, but it won't be just a drug by itself at the present formulation. For that, we need further improvements in the future.
And the final question, a lot of people are asking questions related to the absorption in the stomach, including how is the formulation helping it better absorption through the stomach? There is one that asks, does inflammation in the GI tract of any sort interfere with the absorption in these peptides? And another one which says, what if most of the protease inhibitors are used in the stomach only? Would you like to comment on the interaction in the stomach and the absorption?
I think Avon should answer it.
It happens not in the stomach, it happens in the small intestine. So in the stomachs, we just want to go through it safely. And then for that, I think the formulation of the capsule, the enteric coating is very good. And most things are absorbed not in the stomach, are absorbed in the small intestine. And that is the case also with insulin. Of course, we have to enhance the absorption, and that is done by enlarging the junctions between cells. So maybe that is one thing that could be improved in the future.
All right. Thank you so much. We're at the end of time for the question answers. Now, I would like to thank the panelists again for the valuable insights and the interactions. And thanks are also due to the audience for such wonderful participation. The questions that haven't been answered will be answered through a follow-up email. I would now like to request Dr. Kundu and Dr. Nupur to give their concluding remarks and close today's session, please.
Thank you. Thank you all.
Thank you.
Bye.
Bye-bye. Delight. Bye-bye. Thank you so much.
Thank you, sir. Thank you, Professor Hershko. Thank you, Miriam.
Bye.
Thank you.
Take
care.
Bye. Bye.
Thank you to all the audiences.
Thank you to the audience.
There.
And our next one will be held on December 8th. So please stay tuned. That'll be episode 7. We will very shortly release the information about it. In the meantime, Have a very good morning, a very good evening, and good night to everyone. Thank you so much. Bye-bye.
Stay safe, everyone.
Bye.
Bye.
Bye. Thank you. Bye. Bye. Bye.
Bye.